right now. Um, so we're going to do that. And then on Friday, we're going to do a little bit of enzymes talk. And then we are going to talk about the exam. I haven't looked recently to see who's done the practice exam. I presume that some people have, and um, great. I think that's going to be that's there to be a good practice, um, as described. So with that said, I'm gonna go ahead and get moving from where we took off the other day. So we were talking about sarcomeres and the different components of a sarcomere. And these are your major building blocks of muscle cells and muscle fibers. There's these different zones within a sarcomere that reflect or are reflected by specific proteins. And, this overlapping region is this pair of proteins. And um, all of these proteins that we're talking about here are largely filamentous. There is a little bit of variation there, but now what we're going to get into is some more of the details there. So if you look at this, this is going to introduce up top, we have basically one complete sarcomere, um, indicated by our Z disc going all the way over to our other Z disc. We have an actin filament, or we have an actin filament that kind of looks like you know, a twisty corn cob of sorts. And then in the middle, we've got myosin and all these unique little myosin head groups. So we've got our myosin filament that basically extends from the M line toward the Z disc, but doesn't actually attach to the Z disc, and then on both sides of that M line. Now, the interaction that we have within a sarcomere is between our myosin head groups and our actin filaments. Now, in addition to that, we've got a pair of other proteins that are worth introducing, titin and nebulin. Now, if you ever look up, I'm like 99% sure this is the case. Um, if I remember correctly, Titin is one of the typically largest proteins. I think that there's another perlecan, but Titin is a particularly large protein. If I remember correctly, it is somewhere in the order of like 30,000 amino acids. So it's a massive protein. And what it does is it's going to span from the Z disk all the way to the M line. And one of the things that it's effectively going to do is kind of connect those two. So Titan will extend from the Z disk to the M line and more or less act as a spring to keep those myosin filaments centered on that sarcomere. So it kind of keeps everything in line. Molecules of nebulin, another protein, this other protein, what that's going to do, or what molecules of nebulin are going to do, is basically they're rotated around these actin filaments. Now they extend from the Z disk, and basically they're believed to determine the length of these actin filaments. So now this might be, I don't think this is too much of a stretch, but nebulin, if this, theory or this idea, nebulin, not nebuline, if this idea or theory kind of holds true that it is playing some role in regulating the length of actin filaments. For anyone who's taken a like cell biology class, is there something else like on the genetic level, a complex or a structure that regulates the length of a I don't want to give it away, but I'm thinking about genome level regulating length of I'll just say ends. The ends of and here's another hint. There we go. What I'm thinking of is these kind of caps.
Bye, you too. See you later.
What I'm kind of showing there, trying to draw. Yeah, I am talking about telomeres. Great. So telomeres, they play a role in regulating the and helping prevent like circularization of chromosomes. And so, yeah, that's kind of what I was getting at. So nebulin, I can imagine that if it's playing that role, one of the things that nebulin's doing, or it's analogous to a telomere. Um, okay, so that's what titin and nebulin do. Nebulin's kind of wrapped around actin, and titin extends from the Z disc to M line. And so here what we have is one titin and another titin. So we have two, yeah, there we go. Two titin proteins displayed there. They're all my drawings. Proceed. Now, when we think about these myosin and actin filaments and what make them up, um, actin's kind of a complicated protein, and we have an interaction. What we're really interested in is the interaction between the myosin head groups and actin. Now, the thick filament of myosin displayed here on the right, it's kind of I mean, it's a filamentous protein, but these head groups are really kind of the action, where the action's at, and what's kind of most important and most interesting. But before we get to that, we need to look at actin. Actin participates in many very important cellular processes. Actin is involved in muscle contraction, obviously, cell motility, cytokinesis, or your cell division, vesicle and organelle movement, cellular signaling, so perceiving something from outside of a cell and communicating that inside, cellular shape, so it gives us a cell structural integrity. And these are and one thing that actin's also involved in with this vesicle and organelle movement is I've always kind of thought about, and because this talk that I went to as a graduate student presented actin as almost like a highway. There are proteins that jump onto actin, and that's how they move about the cell. And so with this vesicle and organelle movement, it's these associations with actin that make them fairly mobile. Now, many of these processes are mediated by extensive and intimate interactions of actin with the cellular membranes, for example, the plasma membrane. Now, what actin kind of looks like is a little bit tricky. It's maybe not tricky, but it's not a one-dimensional explanation, or you can't use a one-dimensional explanation, because what actin is a globular protein that binds to ATP. It also binds to ADP. Now, that binding of ATP and ADP is basically your connection between your muscle contraction and movement that you have and actin and myosin interactions. So G-actin in particular is the protein that we're interested in. Now, what G-actin does, its role in binding to ATP is that's how it begins to polymerize. And what I mean by polymerize is not the individual G-actin protein. So imagine that this right here is one G-actin subunit. It will bind to ATP, hydrolyze that ATP, release the inorganic phosphate, and remain bound to ADP. And as long as that's bound to ADP, it will begin to assemble and aggregate with a number of other G-actin subunits. Okay, so this would be an example of a quaternary structure. Each one of these G-actin subunits is bound to ADP. In order for, and I'm going to put kind of check marks along the interface, in order for this aggregation to take place, the hydrolysis of ATP had to happen. So essentially, G-actin had to hydrolyze ATP in order for that to take place, and that to take place, and that to take place. So it's almost like ATP hydrolysis takes place, and that's when we can add another G-actin subunit to this complex. And they assemble in this particular way where it's almost like head to tail. The terms that, or the terminology that's used is the plus end and minus end, however. Now, when we look at one G-actin subunit, like I've got right there, that one G-actin subunit is the form of the protein that has not yet bound to ATP or is not bound to ADP. And that's the form that you're going to find whenever it's not assembled into a large complex. When you look at those individuals, this end right here would be your plus end. That plus end is important because that's the way that it's going to mediate a contact with the minus end of the protein or of another G-actin subunit. In addition to that, that is the end that is ultimately going to attach to the Z disk. So when it comes to the kind of bookends of a sarcomere, it's this end that I've just kind of highlighted in red that is affixed to the Z disk. Now, G-actin forms polymers called F-actin, such that one actin is in contact with four others. Now, that's a little bit confusing, I think, because when you look at this right here, well, I see one connected to one. But what we have is multiple threads of this, multiple of these chains that kind of rotate around one another. So, for example, this, let me clear all my drawings out here. This right here is, or actually, let me shrink that range. Um, this interface right here is like this interface right here, which is like this interface. Okay, so this kind of filament is basically one of these two. You can look at this and say that there's two filaments there. And those kind of wrap around one another to make kind of a thicker filament. So the sometimes when you think about proteins as being filamentous or globular, the idea of a filamentous protein, um, some filamentous proteins that are referenced would be like collagen. Collagen is a filamentous protein, and when you look at it, it's one polypeptide chain. And it's got a very tight turn to it. Actin, on the other hand, is a filamentous protein that results from a globular protein. So it's multiple polypeptides that have folded together. So F-actin is actually going to resemble that double helix. So this F-actin is what's highlighted in this figure D here. Now, polymers have a polarity. That's that plus end and that minus end. And the minus end is kind of that opening. That's going to be where the ATP is going to be bound, where the ADP is ultimately going to reside. The plus end is going to be what attaches to the Z disk. So when it comes to actin, a couple of things that are important with respect to its function are that actin does what's called treadmilling and cross-linking. So treadmilling is the way that something can move across or move alongside a actin filament, and cross-linking is a way that that's particularly relevant to muscle contraction, because things will interact with actin, then release, and then interact with it again. And we're going to get into the specific steps of that in just a little while. So those are the two important functions and kind of capabilities that actin has. So a number of proteins will bind to actin through domains within their sequence that are known as actin binding domains. And this brings up a particularly important point or good point. A domain is basically just a region of a protein that has a kind of known function, or has an apparent function, has a function that's been observed over time. So capping proteins bind at the ends of actin filaments, and some capping proteins stabilize the actin filament while others promote disassembly. So essentially what there are is like they're either going to kind of lock that filament in place or they're going to begin unwinding it, helping those different G-actin subunits release ADP so that that depolymerization can take place. And this statement of some have a role in determining the filament length, one example of that would be nebulin. Now, actin filaments can undergo a process known as treadmilling, where the filament length remains relatively constant, while actin monomers add to the plus end and dissociate from the minus end. Cross-linking proteins organize actin filaments into bundles or networks, and actin binding protein alpha actinin binds actin filaments into parallel bundles, which hold the bundles far enough apart to allow interaction with myosin. This right here, alpha actinin. This is important because essentially what it does, so it holds these actin filaments kind of parallel with one another so that they don't collapse in on one another. So that we have, when we look back at a sarcomere, we have this separation. So we have this space right here. Because if not for that alpha actinin, those actin filaments might not maintain that same structure. So if there's like a alpha actinin that's dysfunctional or something like that, then you would see that kind of collapse or that low proximity between those subunits or between those actin filaments, and that would mean your sarcomere structure is going to be compromised. Okay, so now our other major player here is myosin. So essentially, as a little bit of a review, when you think of a sarcomere and you think about the structures, I think the discs and the line are kind of, I don't want to trivialize it and say easy, but I think that I, they're a little bit less complexity. The ends, your bookends, are your Z discs. What's helpful for me is I think about what's the last letter in the alphabet? Z. Z is at the end. Okay, so Z's are at the end. In the middle is your M line. Why M line? Well, midline. It's the midline of a sarcomere. So there's our ends. Now, what is anchored to my Z discs? Well, those are actin filaments. The plus ends of the actin filaments kind of thread inward. Do they connect to one another? No, they don't. So my sarcomere Z discs are my boundaries, or my Z discs are my boundaries of the sarcomere. I've got an actin filament going to about here. I'll use the left-hand side of my head, and then another one going from here to the right-hand side of my head. So those actin filaments kind of go from my hands to my face, basically. Now, in the middle, my M line, I've got thick myosin fibrils. That myosin is going to have a head group. Now, that myosin head group is particularly important for the contraction and relaxation. Okay, so ATP is essential because ATP helps those actin filaments assemble. Those actin filaments, those all are bound to ADP, which I like this model right here. Sorry to keep going backwards, but I like this model right here. And I like this figure right here because this is one actin filament. Let me pick a different color than red on red. I'm going to go with orange on white. Okay, so this is one actin filament or one actin G-actin subunit. This right here is that ADP that is bound to that. Okay, so what I currently have highlighted in orange or squared off in orange is one G-actin subunit, G standing for globular. That globular protein is bound to ADP. There's my ADP right there. This whole thing right here forms an F element, an F actin for filamentous. So G begets F as opposed to F begetting G. Okay. Um, so that's one of the reasons that I think this is helpful, or I think that this is a good figure here. Now, we go back. Okay, so a myosin protein. Here's essentially a nice little depiction of it. A myosin molecule has two major parts. It's got a head group and a tail. The myosin tail and myosin tails kind of bundled together to give us that thick filament. They have a repeat section. Now, what that means is that the amino acid pattern, or there is an amino acid pattern, and that amino acid pattern is represented by A, B, C, D, E, F, G. So it's these seven residues, and then basically those seven other residues. Now, we have at A and D, we have nonpolar amino acids. Some good examples of nonpolar amino acids that would go there would be something like, oh, probably switching my pen. We wouldn't want to put a serine there, but maybe we could put like a glycine or a leucine. Those would be good examples to go there. Now, the tail of a myosin molecule is a helical coiled coil. So this is a helix right here. Notice the two different polypeptides are represented by different colors. One's orange, one's slightly green, to represent that they are distinct. So this would be an example of a quaternary structure that's folded. Not folded into a globular protein, but this is a filamentous protein or a pair of filamentous proteins that have assembled into this thicker filament. Now, we've got our two polypeptides. The tail represented here to here, which just to give you a sense of how large this is and to put it in context, this is approximately 950 angstroms. Okay. Now, this is a coiled coil with a seven residue nonpolar pseudo repeat. So that at A and D, it's pretty consistent that it's going to be a nonpolar residue, but there might be some variation in those other positions. So B, C, E, F, and G. Those could be polar, those could be nonpolar amino acids. It's going to vary a little bit. And if you were to dissect or look at the sequence of an myosin filament, you might see some variation there, but you're going to see a little bit of a pattern. Now, the head group consists of a heavy chain and light chains. I've always thought that this kind of resembled like an antibody in the sense that an antibody has a was it the heavy chain and the I can't remember. It's been a minute since I've worked with any antibodies. But essentially this heavy chain region of it, that's kind of the sensitive part. That's the part of the protein that has some like apparent function in the sense that it's involved in the binding to actin. Now, another depiction of that with respect to, or another depiction of the head group would be shown here. So here what we're looking at is just the head group. So the basically in both cases, the N termini of the protein. So this right here is depicted in more detail right here. Now, the important parts about that head group that you need to know about. This right here and this right here. That head group, just like our heads, have a neck and a kind of motor portion, your mouth being the motor portion of your head. The neck domain, what it's going to do is exactly what your neck does. It's going to move. The head is going to carry out a particular function, specific function. The single myosin head structure is going to do two things. It's going to bind to ATP. It's also going to bind to actin. Now, what that looks like in terms of its functionality, let's go ahead and jump up to, okay, so here's another depiction of our Z disk. and the movement and contraction, basically those Z discs becoming closer or farther. I'm going to jump over here. So here are the steps in muscle contraction. We have a myosin head group. We've already talked about that. I'm going to go back to the question, the top hat question in just a moment. But we have our myosin head group. One of the things that I want you to notice about that myosin head group is it is bound to myosin. It is not simultaneously bound to ATP or ADP. Now, when myosin is going to bind ATP, it then releases. So this nucleotide binding and the dissociation of myosin's head group from actin kind of happens simultaneously. After that, ATP hydrolysis is going to take place. So to put this into context, when the myosin head group is bound to ATP, it is not bound to actin. It hydrolyzes ATP, and then what it's going to do is that neck domain is going to basically pivot, and it's going to move. So you can see this as a subtle movement right here. That kind of contracts and it shifts and it elongates. So when that ATP hydrolysis, I always think about it like it binds ATP and then it hydrolyzes so that it can release inorganic phosphate or have the ADP and inorganic phosphate bound, so ADP and PI. It elongates. Whenever it elongates, then it basically grabs onto another part of one of those actin filaments or that F actin. Whenever our protein or our myosin head group, which has ADP and inorganic phosphate, shifts and binds to actin, then it can release the inorganic phosphate. Okay, so thinking about this in terms of what it's bound to, it being the myosin head group, if it's bound to ATP, is it bound to actin? No. If it's bound to ADP and PI, well, then ADP and PI, which are different from ATP, then it is bound to actin. When it releases the inorganic phosphate, it's still bound to actin, but it also pulls the actin, which is indicated by this arrow right here. What that means is that our actin filament moves, moves in the direction. This arrow or these two arrows right here are indicating closer to the M line. So if our actin filaments are moving closer to the M line, what does that mean for our Z discs? Add to the chat, are those Z discs getting closer together or farther apart? Are the Z discs getting closer or farther when this takes place? The Z discs are getting closer. You're absolutely right, because our muscle has contracted. Now, that's all because those thick myosin or the myosin head groups have basically bound to actin and pulled it. So it's ATP hydrolysis that enables that muscle contraction. And then once that contraction has taken place, ADP is released because the actin has moved, or that's kind of the sequence, not necessarily because, but actin has moved. Our kind of elongated neck domain, notice how it was elongated right here. When that gets shorter, then we can also release ADP. And this can happen again. Alternatively, what can happen is, well, we can relax. That actin can extend outward. Now, what that looks like kind of on a larger scale is, oh, not that show. On a larger scale, it looks like this. Drawings. Our Z disks have gotten closer together. What this is showing is two adjacent sarcomeres, each with their own M line, each with a pair of Z disks. One Z disk is paired or shared between these two sarcomeres. Our myosin head groups are basically here, 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 here. And when ATP binds, what's going to happen is they are going to um, reposition. They're going to hydrolyze ATP and they're going to bind at a different place on these actin filaments. So if we think about this as, I'm going to put a check mark where they are initially bound, here and here. Okay. Well, those myosin head groups, do the myosin head groups move at all? They do because that neck domain elongates, but from whenever a muscle contracts, there's a good way of looking at it. When that muscle contracts, does that myosin head group, has it changed position from like its start to its finish? Or it's kind of like it runs in a loop in the sense that its starting position is the same as its ending position. Does the so even whenever that muscle contracts, those head groups are effectively, for all intents and purposes, in the same place. The Z disks have contracted, but that's because the myosin head group has bound to actin, it's released, it's bound to a farther place and pulled it. That's effectively what's happening there. Okay, so we have these very small movements, a large number of these small movements that cause this muscle contraction. And if you think about these sarcomeres, all these motions that we have, not just in our arms and everything like that, but your heart muscle contracting and relaxing. This is what's happening. This is our ATP demand. Now, got a question for you. Z disks move away from one another when a muscle relaxes. Let's go ahead and answer that. I've got a good feeling that everyone's going to get this one right. Okay, 10 seconds. Right, let's go ahead and take a look at that. The Z disks move away from one another and the muscle relaxes. That would be true. When they contract, they move close together, but when they relax, they're going to extend, they're going to elongate. Now, here's another depiction of what that looks like. So our relaxation Z disks migrate away, contraction, they get closer. And that's all because of that ATP hydrolysis. During muscle contraction, each sarcomere is going to get more narrow or it's going to shorten, bringing those Z disks closer together. Visually, there's no change in the width of the A band. Both I bands and H zones completely disappear. Oh. And this is caused by actin and myosin filaments sliding past one another. Thus, the actin filaments move into the A band and into that H zone. So if we were to compare this to what we saw basically in our I think it was our second slide about muscle fibers. Now those steps in movement that's taking place. ATP binds, causing a release. ATP hydrolysis takes place, causing the cocking of this, the kind of contraction or the cocking of that head group there, or that neck group, followed by a weak binding. The inorganic phosphate, the PI, is going to be released, which causes strong binding, followed by a power stroke or that movement of that actin filament. ADP is released, And that movement causes the movement of our actin filament causes ADP to be released. Now we'll tackle one more question, and then yeah, we'll look at that. If myosin is bound to actin, and I think that this is one sort of question that you should be thinking about with respect to the sequence of events. Think about what's bound to what at a particular point. If myosin is bound to actin, is myosin bound, sorry, that should say bound to ATP? Is myosin bound to ATP? For this, our correct answer is no, it is not. Great job, everyone. Okay, so essentially what's shown in the next few slides is kind of another depiction of that sequence of events. I like this model because it shows that um, in this particular cell right here, it shows our movement of that. In addition, our movement of that myosin head group. In addition to that, if you look closely right here, you can see that ADP, or what's intended to be shown there, is that we've got ADP and inorganic phosphate in the nucleotide binding site as opposed to ATP. Here, what we see is that hydrolysis is going to take place. That's going to require water. And then we're going to kind of see our migration and the shift of our actin filaments. Now, ATP binding causes a release. Hydrolysis causes the cocking followed by the weak binding. Inorganic phosphate causes that, or release of inorganic phosphate causes the power stroke of our actin filaments. And essentially these, let's see. Yeah, those are the important things that I wanted to show with respect to that. Now, I think what we're going to do is we're actually going to pick this up on Friday because I feel good about where we are. Um, so I'm going to stop my share and stop my recording.